Okay, we are recording.
Awesome. Thank you. Good morning, everybody. Thanks for joining this second water issues seminar of February. Almost at the end of the month here. I had to stop and think while I was talking about the date and time. Yeah, we've got Dr. Jake Walsh with us from the University of Minnesota to talk about some carp management projects. Um, Dr. Walsh is an assistant professor at the U's Department of Fisheries, Wildlife, and Conservation Biology, and he studies lake ecology and stewardship, especially the understanding and managing of aquatic biological communities and ecosystem services with state, local, and tribal partners. So relevant to a lot of our work. Prior to joining Fisheries, Wildlife, and Conservation Biology, Jake worked at the DNR as an aquatic invasive species research and grants coordinator, or maybe the aquatic uh, invasive species research and grants coordinator. So thank you for joining us this morning. And I'll go ahead and let you take it away. And in case it's weird and folks didn't catch this, Jake's audio wasn't working on the computer, so he is speaking through a second Jake Walsh, but his video is on a different one. So just wanted to give a heads up on that.
Yeah, yeah, thanks, Fox. And sorry for the technical difficulties. And yeah, hopefully it's pretty straightforward. There's only one Jake Walsh on the call, and I think that's a good thing. So thanks so much for having me. I'm excited to talk about this. This is a talk I've given a couple times over the last year, but it's a really fun project that we did in partnership with the Minnehaha Creek Watershed District. So Brian's on the call now. Jill Sweet might be joining us later. But yeah, we're looking at carp management projects in the state. And I think that's something that this group may or may not be directly familiar with, but as a water quality management tool, I think it's something that comes up quite a bit in Minnesota. All right. So like I said, this is a really awesome research team. So I partnered with, I was a postdoc at the time working with Dr. Dan Larkin at the U. We also worked with Jill and Brian at the Minnehaha Creek Watershed District. And then a good group of pollution control agency staff were a big part of advising on this project as well. So big thanks to Jeff, Amy, and Allison. Also for Allison providing data, not just for this project, but I feel like either directly or indirectly, I'm always asking Allison for data, and I really appreciate that connection and that advantage we have in Minnesota. We also worked with the Minnesota DNR and Brian Nirbon in particular. And the work was funded through the Minnehaha Creek Watershed District, which made this feasible. So one thing I like to start out with here is this NALMS paper from Jeff and Amy, thinking about what leads to lake improvements. Like how do we get lakes off of the impaired waters list? And they reviewed 20 years of lake nutrient impairment delistings in Minnesota. And they found basically there's no quick fix or silver bullet activity. I think that's something that's probably not super surprising to anybody on this call. But I think it's also really important context here. They found that external load management was really critical, where 93% of lakes delisted over that time period involved some sort of external load management. And then extending from that, pairing external load management with in-lake management is also a really important activity. And so 51% of delisted lakes had that sort of general approach. And I think that's a really important part of how carp management is done. So I like this figure that we put together just because I think it shows some of the complexity. It's oversimplified in a lot of ways. But you can see carp here. The arrow is pointing down into sort of my simplified lake and how they impact water and floristic quality through disturbing plants, disturbing sediments, which can lead to algae blooms. But it's in this context of broader lake management goals. So we, the broader management community, or I still say we a lot. I guess I'm more on like the research side of things now, but my time at the DNR is kind of sticky in my brain, are doing a lot of other things in lakes. So yes, we're directly managing carp populations, which is what we'll talk about today. But we're also managing water quality directly through alum treatment, through aquatic plant management, and then maybe even more importantly, like we talked about with those delistings, we're managing the watershed and trying to reduce the total nutrient load into the lake. And so I think one way to think about this work is carp management as a tool in the toolbox, and thinking about lake management as a much broader and more complex activity is helpful for thinking about carp. And so this sort of brings us to some of the work that was the impetus of the research that we did. And so the Minnehaha Creek Watershed District, they were working on restoring lakes within the 6 Mile Creek Halstead Bay sub-watershed that they manage. And carp removals were a big part of that. And so that's sort of what spurred a lot of this work, because they were finding overall pretty successful carp management projects in these lakes. So they installed barriers, they removed carp directly. You can see here in the before picture on the left, there's a lot of lakes that are those more warmer colors, indicating higher carp biomass. And then in the after, a lot of the lakes are these much cooler colors, indicating lower carp biomass. So there was a pretty significant reduction in carp biomass in many of these lakes. And what they saw was a relatively mixed lake response. So here I have two different ways of looking at this, or Jill actually put this together. Jill has two different ways of looking at this. One is the summer occurrence of plants in the littoral zone, and the other here is total phosphorus. And for both of these, you're just looking at before and after the carp removals. And you can see that in some cases, these lighter blue colors, you see the responses you would expect in carp management projects. You're seeing increases in littoral occurrence for vegetation, decreases in total phosphorus, but that is also, but also they saw some more varied responses as well, where there were some lakes where vegetation didn't change much. You'll note that a lot of these lakes are lakes where there was already a lot of vegetation, above 50% of the littoral zone. And then there were some lakes where you didn't see as strong of a response in total phosphorus reductions. And so this variability was the core of some of the research that we did. We wanted to quantify how variable lake responses were to carp management, but do that at a broader scale, trying to bring together as much statewide data as we can to think about that variability. And then we wanted to start thinking about what drives that variability, and then also what are the important ecological relationships in carp managed lakes. I'm thinking about drivers, thinking about relationships. The core there is really thinking about how do we use the information from this research to inform management? And so one of our core goals throughout all of this is to set data-driven guidance for managers and stakeholders, trying to think about when is carp management a useful tool? When do we need to reach for other tools in the toolbox? And so our approach was largely bringing together big statewide data sets, which is something that I really love doing. So if you have projects that are related to data and related to lakes, I'm always happy to chat. We got water quality data from Allison, from all of you. We used our Minnesota Aquatic Invasive Species Research Center PI charter database, which has over 3,000 aquatic plant point intercept surveys from lakes to look at aquatic plant responses to carp removals. And then Jill put together a pretty large data set of carp biomass and removal estimates from local managers, contractors, and lake associations. So the people out there actually doing the projects. And so before I move on, I do want to say a lot of the research that I do, and I think a lot of the research that needs to be done in lake management depends on the statewide commitment to data sharing and monitoring. And so I just want to just emphasize how valuable it is and how grateful I am to be in a state where like this work is actually fairly achievable at this statewide scale. So thanks to all of you for the work you do supporting EQUIS and supporting some of the other work that we do in lakes. What we ended up with, here's just a bar graph of the number of lakes with different types of data. We ended up with a good number of lakes with carp removal data. So we had 90 lakes where we had some kind of carp removal data. But then as you start to overlay these other data sets, thinking about EQUIS and water quality, thinking about PI charter from MACIRC, you start to lose lakes where you have overlap of all of those different variables. And so what we ended up with was 15 lakes with pre-post data for all of the lake response variables we were interested in, which feels small relative to the 90 lakes that we started with. But one thing I've always mentioned alongside this presentation is like, if you were to go out and try to design a field project where you were looking at 15 carp removals or carp management projects, that would be millions and millions of dollars. So this is still extremely valuable data, even if sort of the Swiss cheese of layering all these data sets together led to a relatively small number of lakes than what we started with. And one other thing I want to mention is one of my goals here was try to use as much of the data as we had whenever possible. So as we're looking at like change in a system, just because we don't have plant data doesn't mean we can't look at change in water quality. And so that is one thing to keep in mind as we're going through these results is it's not the same subset of 15 lakes in every analysis. Um, there are some lakes where we have more water quality data. There are even some lakes where we have plant data but no water quality data, and those would be included as we're looking at lake responses to carp removals. Okay, so we use this core data set to think about variability in lake responses to carp management. And so just to provide some expectations here, when we're thinking about carp management, we often have criteria that we use to evaluate whether or not they're successful. Obviously, we're looking to reduce carp biomass in an effort to reduce nutrient concentrations in the water column, improve water clarity, and then through both improved clarity and reduced disturbance from carp, lead to more healthier biological communities. And healthy biological communities is a pretty subjective term. Here, we're just looking at macrophyte coverage in the littoral zone. So how many aquatic plants are in the littoral zone in the lake? And to show some of this variability in lake responses, I'm going to present a lot of figures that look like this. So here, it's a pretty simple plot. You just see on the y-axis there, you have change with removal. If it's a positive change or an increase, you can see it's above that 0 line. And if it's a negative change or a decrease, it's below that 0 line. And of course, we want some variables to increase, some variables to decrease. And so I have this shaded blue area to show like what the desirable outcome is. So in this case, we're looking at a variable where we want to see a decrease in carp biomass. And so this particular example is carp, or sorry, in a variable. And so this particular variable is carp biomass. The goal here is decline. We're looking at kilograms per hectare, so just the biomass density in a lake and how that changes with removals. And so I'm going to present a few different things on these figures. First is just the decline in the median lake. So thinking about that 50th percentile, how did that particular lake respond to carp removals? Here you see that median lake saw a decline in carp biomass. The other thing I want to present here is just the range. So here, these lines will represent 90% of lakes. So 90% of lakes had a carp removal of it, I guess at least a carp reduction of at least 0 kilograms per hectare, just above 0. And then all the way up to roughly 500 kilograms per hectare. And so on average, not on average, in most lakes, we're seeing a reduction in carp biomass. And the last little piece of data I put on these plots is an effect estimate from a mixed effects model. If you want to read the report, there's a lot of benefits and caveats to this approach. But for the purposes of this talk, just think of it as the average sort of effect of removal on the given variable. And so here you can see we have an estimated effect of minus roughly 220 kilograms per hectare of carp, which is a pretty good reduction, and that's what we want to be seeing in these models. And so looking at water quality, you can see that generally the changes are matching our goals. We're seeing declines in total phosphorus in the median lake, declines in chlorophyll A, measure of algae, and then an increase in clarity. But one thing to note is that this is within a pretty huge amount of lake-to-lake variability. You can see that 90% of lakes in each of these cases crosses that 0 line, where there are some lakes that are responding or not necessarily responding, but changing in the opposite direction, which is one thing that's really important to note here is that we're really just looking at change over that time period. Lakes are dynamic. There's lots of different things driving these variables. And so it could be that something else is happening in that lake, or even how we're measuring these data is just a little bit messy. And so there's a lot of reasons why we see variability, which I think I'll talk a little bit about after we get through these plots too. But when you look at these average effects, you can see that generally, there is a pretty consistent negative effect on total phosphorus, a pretty consistent positive effect on water clarity, which is what we're looking for in those cases. But that chlorophyll A effect is somewhat unclear. And again, I just really want to highlight that lake-to-lake variability in these responses. Thinking about aquatic plants, again, here across all of these different metrics, we're looking for an increase. So I have a couple measures of aquatic plant diversity, just overall macrophyte richness, and then also floristic quality, which is a measure of um, sort of the disturbance tolerance of lakes, or like how conservative those plants are. So thinking about plants that don't do particularly well in highly disturbed systems would have a high floristic quality index score. And then the last metric here is just submersed frequency of occurrence in the littoral zone of lakes. So that's thinking about the overall coverage of aquatic plants in lakes. And you can see here that, yeah, the median lake generally had a positive response in each of these three variables, but there was extremely high variability in the plant response in particular. And this came through in our mixed effects modeling as well, where we saw the desired outcome in those diversity metrics and richness and floristic quality. We saw roughly one additional species on average, and an increase in FQI of about one, but the occurrence or coverage response was somewhat more unclear. And again, I just really want to highlight how many different factors influence macrophyte occurrence in lakes. But there is a lot of variability here. And so one thing that I think is worth digging into here a little bit is that a lot of the pilot studies for carp removals highlighted plant recovery with carp removal. A lot of these lakes were relatively murky and turbid with very low plant populations in these pilot studies. And what we're seeing here is somewhat inconsistent with that. And so I want to emphasize just the breadth of the kinds of lakes we're looking at. And so here I have a plot of frequency of occurrence of plants in the littoral zone here on the x-axis, and then the change in the frequency of occurrence here on the y-axis. And so one thing you can see here is there's a negative relationship where if you have very few plants before the carp removal, that's where you see that potential for an increase in vegetation. But if you already have a vegetated lake, that's where you see more of that mixed response, where yes, there are some lakes where you still see a very positive response, but it's more of a mixed bag there as you get above like 50% frequency of occurrence in plants. And so we had a lot of already vegetated lakes in the study, and that might be part of why we're seeing such a muted overall response in lake vegetation communities. The other things I wanted to mention here quickly are just thinking about where variability comes from. This is a somewhat opportunistic data collection effort. I was trying to be really open about the kinds of projects we brought in, not just to bring in more data, but also to reflect the full breadth of these projects and the full breadth of how these are done and how these play out in the state of Minnesota. And so what this means is we have a pretty broad range of projects. A lot of these might not be textbook carp removal lakes. We have varied lake conditions, like we have some lakes that are really well vegetated already. We have varied project scopes and successes, where some of these were really big investments in carp removals. Others were somewhat smaller based on limited resources and capacity. And then, of course, we also have varied monitoring schedules. Some of the data that we collected wasn't collected for the sole purpose of measuring the carp response. And so like a pre-measurement could be a couple years before the carp management actually happened, or like a post-measurement could be a couple years after the carp project was done. Okay, and so I think one really important question to think about this is trying to think about what's driving this variability, because this will tell us, I think in large part, how we can be more selective with our carp management projects and better use that as a tool in our toolbox for lake management. So here I have a pretty preliminary analysis. I think this is where that 15 lake sample size really comes into play. So I have a pretty simple linear model approach to thinking about drivers, which also means, because of the small sample size, we have relatively few drivers we're evaluating right now. But I was looking at these 5 variables for lake responses. So total phosphorus, chlorophyll A, water clarity, and then aquatic plant diversity and aquatic plant occurrence. And then we were thinking about drivers related to the carp management project. So thinking about how successful was the project in terms of percent biomass reduction for carp. And then also, I guess, kind of a measure of how challenging it was of that pre-removal carp biomass, if that maybe had an effect on how successful these projects were. We also had we're starting to scratch the surface of lake characteristics that might be driving project success using lake depth. And then one other thing that came up is we had 4 lakes of these 15 where there was a coincident alum treatment that happened roughly at the same time as the carp removal. And so again, this is a very small sample size, but you might expect pretty strong effects on clarity and algae and total phosphorus with those alum treatments. And that's something we included in these models as well, even though it's just 4 lakes. And so what we found is that we saw that a more successful reduction, so a higher percent biomass reduction, and a lake being shallower led to more positive responses, positive being desirable in total phosphorus and chlorophyll A. So those were reduced more in cases where you pulled a lot of carp out or cases where you were working in a shallower lake. Which is not super surprising. A lot of the work that was done on carp removals initially were done in shallower lakes. One thing that was maybe not surprising, but really interesting was that a coincident alum treatment also had a really strong effect on water clarity and aquatic plant diversity. And so again, this is 4 lakes, but I think this is something that could be a really interesting future research direction, is to look into how alum and carp management work together, either like synergistically or maybe they're unrelated. But I think there's something worth unpacking here, in large part because a lot of these projects, they're not designed for this careful experimentation. They're designed for meeting goals and meeting water quality goals. Okay, and so I think one thing that I wanted to dig into a little bit more based on this alum response was thinking about the importance of integrated strategies. And so I put together a really complicated figure, and I tried to simplify it quite a bit here to look at 3 more or less like integrated strategies. Integrated just being like sort of these multiple approaches to managing lake water quality. And so I have boxes here, and the y-axis of this box, I just have lakes sorted where the most positive responses across all of our metrics, so water quality and plants and carp, are on the top, and then the least positive responses are on the bottom. And then I have that across 3 different strategies. So whether or not there was an alum treatment during the carp removal, an alum treatment that happened sometime before the carp removal, or whether or not this lake has a TMDL plan. And so one thing I noted here is that 4 out of the top 5 responses included an alum treatment before or during carp removal. And then 4 out of the 5 top responses also included an approved TMDL plan. So again, this is just starting to scratch the surface here. It's not a ton of lake, but I think it does sort of highlight the importance of trying to tackle these problems with a lot of different approaches that are going to depend on ecological context. Other drivers that we were interested in, 4 out of the top 5 were relatively shallow and littoral, and then all 5 of the top 5 had successful carp reductions, ranging from like a 60% all the way up to a 95% reduction in carp biomass. So again, just kind of reinforcing some of those results we found about drivers. Okay, so the last piece here, thinking about ecological relationships in carp managed lakes. I think one thing that I also want to highlight about this is that I think this is a nice way for us to test some of the hypotheses we have about carp and how they work in lakes and how removing carp should work to benefit lakes. And so my overall approach here was just to sort of draw out the interactions that we think are happening in lakes, both direct interactions. So I have these as more of these like thinner solid arrows as direct interactions between all of these different variables. And then also through some of these indirect pathways, which I have these more thicker, transparent arrows. So you can see here, carp has direct effects on plants, on clarity, and on phosphorus, but it also has indirect effects on clarity and plants through phosphorus, algae, and then clarity to plants, or through its direct effect on clarity to plants. So there's 2 different indirect pathways here that we're dealing with. And to tease some of these effects apart, I use structural equation modeling. I'm not going to get into a ton of details of that, but the idea here is to use multiple regression, multiple regressions across this system of sort of hypothesis-driven linear models. So you can imagine all of these arrows are like coefficients in a linear model. And the structural equation modeling allows us to sort of consider all of these interactions simultaneously, to quantify some of these direct and indirect effects um, in the system, which is pretty powerful, pretty cool. One thing to note is it is grounded in correlation. And so if we aren't capturing important relationships here, that's where some of these models can start to not necessarily break down, but need to be interpreted carefully. But I think there's a lot going on here that we are able to capture with with this modeling approach. So we split the models into um, an early season model and a late season model. So the early season model is looking at how are these relationships working from April through June. So thinking about that early season, um, algae blooms, external loading, things like that. And one thing that I've noted here is that all of these results pretty much support our hypotheses of how direct and indirect paths should be working in these systems. So you see strong direct effects of carp on total phosphorus, a positive effect there, a direct negative effect on clarity. Oh, I'm pointing at the wrong screen. Direct positive effect on phosphorus, direct negative effect on clarity, um, and then a direct negative effect on macrophytes as well. We also see that these indirect pathways make a lot of sense as well, where carp positively impacts phosphorus, which positively impacts chlorophyll A in algae, which negatively impacts clarity. And then clarity has a positive effect on macrophytes. Macrophytes need light to grow. One of the big reasons why carp are so destructive is because they impact clarity and impact plant growth. So this early season model fits a lot of what we expect. Oh, and also our sort of measure of carp harvest had a strong negative effect on carp biomass as well, which you would expect. When we go to the later season model, so this is sort of midsummer dynamics, July through September, you can see that there's a very similar structure here. You see a pretty strong negative effect of carp on plants. Um, a lot of the same sort of indirect pathways are similar as they were in the early season model, but there's a couple exceptions here. One is that later season clarity is much more strongly affected by algae and actually not significantly affected in the models by carp biomass. So there's a shift here in carp sort of being a co-driver of clarity to really just that algae, in lake algae, being the primary driver of clarity. And there also that indirect path was also weaker in this example, where this direct relationship between carp and phosphorus is about half the size of the early season model. So that's a much smaller effect as well. So that's a lot of arrows. It's a lot of stuff to tease apart, even though I definitely oversimplified that system. Um, but I think there are some things that we can pull from all of these results. And I want to spend a little bit of time going through what I see as some of the interesting research results here. And then if I have time after that, I'd also like to spend a little bit of time thinking about how Jill and Brian and Minnehaha Creek have been sort of conceptualizing some of this. And so just to bring together all of the findings from the research component, I do want to again highlight that variability. There's a lot of variability in lake responses to carp removals. We looked across a lot of different projects, a lot of different lakes, but I think a lot of that variability is likely predictable. We're able to start to scratch the surface with the 15 lakes we had, um, but I think with more lakes and more data, we could be much more prescriptive in how we use common carp management, and we can use that to refine decision making. So a couple examples here: water quality improvements were variable, but again, in lakes where we were able to reduce carp biomass significantly, and in shallower lakes, we saw more positive water quality improvements through phosphorus reduction and clarity improvement. Um, the plant responses were really nuanced as well. But again, that could be largely context dependent. In lakes where there's already plenty of plants, you might not see that same positive plant response. Whereas if you're in a lake that's really low in vegetation, really high in carp biomass, there seems to be a high likelihood that carp is driving that. And those lakes tend to have more positive responses when you pull out the carp. And the other thing that I wanted to highlight here is this importance of integrated management, thinking about lakes where we are not just tackling carp, but we're also tackling internal and external nutrient loading through either alum or TMDL planning and some of those watershed practices. And the other thing that's important here is the seasonal difference that we highlighted with those structural equation models. We saw the strongest effect in the early season, and I think there are a lot of cases where we see lakes where clarity improves really well after you pull in the spring, after you pull out carp, but that summer clarity is still not as positive as you might think it would be based on that early season. And so I think this is where that other management might be really important, thinking about internal loading, thinking more in the long term with external loading as well. Okay, so we're at 9:36. Awesome. I think we're doing well on time. I sandwiched a lot of stuff together right for this talk, and I'm glad that we're on track time wise to still have some time for questions. And so I wanted to pull back to this slide from the 6 Mile Creek, Halstead Bay subwatershed lakes, where we're thinking about Minnehaha Creek Watershed District has a handful of lakes. They have some lakes where carp removal's gone really well, some lakes where it hasn't had as strong of an effect as they were expecting. But they also have many, many other lakes in their watershed that they need to manage. And so how can we use this research to support some of the work that they're doing? And Jill and Brian spent a lot of time putting this all together. I'm just going to be basically reading through these slides, but I just want to highlight that this is like largely Jill's work and Brian and the district putting a lot of these ideas together. So thinking about these management implications. So from the research, we saw the strongest effects in shallow lakes. We saw strong effects when they were part of integrated watershed strategies or in lake management. And we also saw stronger effects with larger sustained reduction in carp biomass. And we saw those larger effects in spring. And so these are the things that are going to be driving how Jill is making decisions about whether or not to use carp management in some of the systems in their watershed. And so here is the broader Minnehaha Creek watershed, or the broader watershed that the Minnehaha Creek Watershed District manages. You can see it includes Minnetonka, also a lot of other metro area lakes. And the key questions here are, should we manage carp? And can we manage carp? So thinking about the lake context for both how we expect it to respond in terms of water clarity in plants, but also how we expect that carp population to respond. Can we actually go out there and make a meaningful reduction that's going to lead to a positive lake response? And so thinking about the should you manage carp, Jill came up with this really awesome just checklist based on some of the work we did, and then also based on some of the expertise that she and Brian have in some of these projects, and based on like the broader literature of carp management. And so the first check here is the lake is degraded. Second check is whether or not some of those other system drivers have been addressed. So thinking about, is there still a very large external phosphorus load into these lakes that might inhibit or lead to a smaller carp removal response? Or is there still a pretty large internal load that again would limit the carp response that we're seeing? The other check is, does the lake have carp biomass? There's lots to suggest that there could be a threshold response here where lakes with very high carp biomass are more likely to be degraded by carp. And then whether or not the lake is shallow, which again was supported by some of our research. And then the second list of checkboxes here is grounded more in, can you actually manage carp? And this is maybe where we're really mixing in some of Jill's expertise with some of the results that we had. So thinking about blocking migration, making sure that carp can't reach spawning areas or can't reseed a population once you pull a population out of maybe a specific bay or a specific lake that's connected to another carp lake. Suppressing recruitment, and then also thinking about whether or not removal techniques are feasible, both in the short term, but also in that long term of like, can we do it? And then can we keep it at a low biomass over time? So Jill came up with a couple really great case examples for that contrast some of these different check boxes and criteria that we can use to think about whether or not a lake is a good candidate for carp removal. So using Jennings Bay in Lake Minnetonka, and then Mud Lake, which is in the 6 Mile Creek Halstead Bay subwatershed. So thinking about should you manage carp? First, looking at this, total phosphorus over time has been well above the state standard for Jennings Bay. Sorry, we're talking about Jennings Bay here. So yes, the lake is degraded. That's a check for that box. But Jennings Bay has a pretty large external watershed load that they're still working on trying to reduce. So that particular system driver of water quality and of plants hasn't been addressed yet. So you don't see that same checkbox there. Oh, I got some animations here that are a little out of order. Apologies. And there's still some evidence that there's a pretty strong internal load in this lake where you have really good clarity in the spring, but then much poorer water clarity in summer, possibly from that internal loading of nutrients into Jennings Bay. And then also from this budget, the internal load is relatively large. The lake also has relatively high carp biomass. And so that's a check in favor of potentially doing carp management in this lake. However, the lake is not particularly shallow. It's almost 60% littoral, which is maybe high in the grand scheme of lakes, but not meeting that criteria of a shallow lake. And its max depth is 26 feet. And so you can already see, like, just based on thinking about the potential for this lake to respond positively to carp removals, that you're not checking a lot of these boxes. And then when you think about whether or not you can actually remove carp from Jennings Bay, that's where a lot of challenges come into play as well. Being one of the bays of Minnetonka, people want to be getting in and out of the bay. You can't block migration across a 700 foot, I think 700 foot or maybe meter, one of those two, a very large stretch of water that connects the bay to broader Lake Minnetonka, which is also going to impact suppressing recruitment because there's going to be sources for carp to have spawning habitat and connect to some of the different spawning habitat in the lake. And then even though removal techniques might be feasible, you could pull carp out of that bay. There would be the broader population in Lake Minnetonka that would be continually seeding that lake or that bay to sort of make sure that carp is maintained through time, and it's not a feasible project in that case. Okay, so in summary, the lake is degraded, carp biomass is high, but there are other factors that still need to be addressed in Jennings Bay before you might expect to see a strong carp response. It can't be feasibly isolated from Lake Minnetonka. And so that because of that, carp removal would be relatively short-lived. And so as a result here, the management recommendation isn't necessarily reach for that carp tool right away. It's continue to address the watershed load and continue to manage the internal load in the lake. And then, of course, as you're going through here, monitoring change to think about, are we moving closer to a point where carp management might be reasonable, given sort of that context in Lake Minnetonka. Okay, so the second example here is Mud Lake. This is again a lake in the 6 Mile Creek Halstead Bay subwatershed. And it contrasts somewhat with that Jennings Bay example. So thinking about should you manage carp, the lake is degraded. Total phosphorus has been above the state standard for quite some time. System drivers, Mud Lake is really low in this watershed, and so it's sort of an uphill battle trying to reduce the external load. But there has been a lot of work done in the subwatershed to reduce external loading into these lakes. And so while it might not get a checkbox here, it's something that I think the district is working really hard on. And there's some evidence to suggest that the internal load, at least, is not going to really dramatically impact clarity in this lake, with sort of the caveat of they need to keep working on that, that external load into the lake. And then the lake is also has a lot of carp, so suggesting a lot of potential there for lake improvement should that carp be removed. And the lake is quite shallow. It's 100% littoral, has a 5-foot max depth. Um, so based on the research we did, this could be a good candidate lake, just based on that. The other really important thing here is that because of where it's situated in the system of lakes and wetlands and streams, migration can be blocked. Recruitment can be suppressed because of that, and blocking access to those nursery habitats. Removal techniques are feasible, and because it can be blocked from source populations, that management can be sustained long term. And so thinking about all the check boxes here for Mud Lake, we're looking at a much more favorable situation, and much better ecological context for seeing successful carp removals. And so the recommendation here, because it's degraded, because carp biomass is high, and while watershed load still needs to be addressed, it's checking a lot of these other boxes that might lead to success in terms of being shallow, having a relatively low internal load, but then also having a lot of potential for successful carp biomass reduction, not just now, but in the long term. Carp management is likely to be meaningful in this lake. And so here the management recommendation is, yes, continue to address that watershed load, but also maybe managing carp here could be somewhat successful. And what they found in this case is as they removed carp, as they blocked carp access to the lake, you can see here that gray polygon is carp biomass over time. They see this positive response in vegetation. So vegetation here, you have spring vegetation in blue, summer vegetation in that darker blue, and this is just occurrence in the littoral zone over time, where you can see recovery in this plant community as you remove the carp and as the vegetation starts to rebound in the lake. And they also are starting to see reductions in total phosphorus with the carp removal as well, and even starting to inch towards that state standard again in more recent years. So Jill did a really great job of summarizing some of these conclusions here, thinking about how does our research actually apply to the real world. So I think that carp can be a really important tool in the toolbox. And I think there's some good evidence for that in the Minnehaha Creek Watershed District. But it might not necessarily serve as a standalone strategy for improving lake water quality. And there's a lot of variability in lake responses, but we can start to think about reducing that variability in how we prioritize lakes. And so Jill explored thinking about looking for lakes with a low watershed load, with a low internal load, so thinking about that integrated management strategy, thinking about the fact that there's no silver bullets, but also thinking about potential for carp biomass reduction. So high carp biomass, low connectivity to make sure that the carp removals are sustainable in the more long term, and then also lakes that are shallow, grounded in some of the results that we saw in our research. So that is all I have to present for you today. I have my email here. Also, I have some QR codes. One will bring you to our partnership project page. We partnered with MACERC in order to host some of the information here. And so you can read about the project there. You can also look at our report. We have a 50-something page report on this that you can find at this z.umn.edu/carp report. Or you can just scan this QR code to get there as well. So thanks for listening, and happy to answer any questions that have come up. I did see some chat going on.
Yeah, there are a couple, excuse me, a couple questions in the chat. Thanks so much, Jake. That was a wonderful presentation. Okay, Maggie asked, with many/most carp IPM projects, it is done in addition to upstream or in-lake treatments. How did you isolate the carp removal outcomes in the lake studies from other management activities that might have been going on?
Yeah, that's a great point. And I think the short answer is we didn't in a lot of cases. One of the things that we try to caveat is like there are a lot of things going on in these lakes all at the same time. It's just part of how we manage them. So in cases like the vegetation response, one of the reasons we maybe wouldn't see a vegetation response is because people are anticipating an increase in vegetation, and they're maybe going out, at least in cases where you can manage invasive plants like curly leaf pondweed or Asian water milfoil, going out and managing that alongside carp removals. And so I have a data set I'm putting together for invasive aquatic plant management data that I would love to throw at this and see if we can see some of that effect going on. But other than alum treatments and then just like the very coarse metric of have they put together a TMDL plan, we didn't try to account for some of these other in-lake management activities. And so that is something that like is definitely contributing to the variability that we're seeing in lake responses, I think.
Awesome.
Yeah.
Okay, next question. Brianna asked, oh, sorry if I missed this. Did you do any bootstrapping or try to fill in gaps in the data with other modeling options?
That's a great question. I didn't, just because the sample size we were working with was relatively small. But I think, yeah, I think that's something that could be worth doing. The other reason I haven't dug too deep into that is because I think there is good potential for more data coming in soon. One thing we noticed is like a lot of the projects we were looking at have happened in the last 5 or so years. And so some of the lakes, we have really great pre-management data, and we know management is happening, but there just hasn't been a chance for post-management data to come in. And so like, I think in large part, I mean, one, this is a 1-year project that we did over the course of a couple of years. And so we just didn't have a lot of time to dig into the stats, but also I think this should be sort of a jumping off point for questions like that of like, how do we more carefully address some of the driver questions with a bigger data set?
Awesome, thank you. Okay, one more question in the chat. Folks, feel free to drop those in there, or if you want to raise your hand and ask your question out loud, feel free as well. But extremely valuable to Watershed and lake managers. What's next for you and your research team? Do you have similar studies you're currently working on or hope to work on? And how can we support more of this work?
That's such a generous question. I am like I said, I'm always up for talking more about this kind of work. I right now am very spread out in sort of the in-lake management world. We're doing a lot of work on aquatic plant management, trying to think about how do we better centralize aquatic plant management data, so we can actually think about the effectiveness and non-target effects of that work. We're thinking about better leveraging our state zooplankton data. My background is largely in zooplankton and pelagic food web dynamics, and that's always been a dream of mine, is to come back to Minnesota, where I'm from, and apply some of those skills to Minnesota data. And we have amazing zooplankton data here. And we also have a lot of projects. So a lot of projects related to that in-lake management. For this carp work, I think what's next here is thinking about how do we create some of those like long-term sustainable data pipelines to think about like, okay, how do we keep collecting management data from the people doing carp management projects? I think there's a lot of potential there. And that's something that I'm always open to talking more about. It was a fairly massive effort from Jill to collect those data, but I think now that we have a framework down, I could envision that being more streamlined and easier. The biggest thing is just continuing to collect water quality data, continuing to like guard and defend those programs. I think it's just, they're so valuable. So for me, like, you know, I anticipate leeching off that data for the next 30 years or so. And so as long as there's great monitoring data going on, and also these like innovative efforts to collect citizen data, which I really love about MPCA, I think we'll be in really good shape. I do have some very specific ideas. And Jeff, I'll probably follow up with you over the next week about an LCCMR idea in particular. But yeah, like I said, a lot of really cool stuff in this area, and I'm really excited to like keep getting it off the ground. I've only been in this position for well, I guess less than a year. I started in the fall, but I had some ramp up time as a postdoc to sort of get things started. So yeah.
All right, we've got a compliment from Amy Tim in the chat. Great presentation, Jake. Nice job, Jill. Great to see the addition of the management based on the research. The checkboxes are easy to convey to the communities who are interested in management. Creates a great tool to have these conversations and decide where to put efforts. Thanks so much, Amy. And then a question from Matthew. Could these tools / analysis be flipped to review potential alum treatment effectiveness?
100%. And that is actually something. So Jill is currently working on a master's with Dan Larkin and I. And so she's looking at how do alum treatments impact plants in particular. Also, Katie Pollock at the U, she's a PhD student with Jock Finley, and she's looking at how water quality responds to alum treatments. And so really interesting, like long-term perspective there of like, in particular, some differences in how phosphorus and nitrogen respond after alum treatments. She's seeing phosphorus tends to stay down longer than nitrogen. And so she's really interested in thinking about nitrogen cycling in these lakes. We're thinking about putting our heads together to think about like comparing alum to carp, because they're somewhat interesting in terms of like the slight differences they have in terms of how we expect lakes to respond to them, even though they're both there for water quality response. Yeah, I think it's that is a really great question and a really awesome research area that I'm excited to keep trying to get into.
Fantastic. I have a question, and maybe this is kind of a high-level silly question, but I work in the remediation division, so I think a lot about disposal. And when I hear about all of this carp removal happening, I can't help but wonder, what is the kind of total mass of carp that is being removed from Minnesota lakes, maybe, I don't know, in a summer, in a year? And what happens to that? Is there anything productive we can do with it? Or I'd be curious to hear about that.
Yeah, that is an active question. And it was a major challenge. Like I remember seeing this when I was at the DNR. That was one of the big concerns, was like, what do we do with these carp? Because if you're harvesting them under a research permit, you can't do much with them. Like you can't sell them. And so you're left with options of like, could we feed birds at the Raptor Center? They are looking at like digesters, like digesting them into fertilizer. I think there's some cases in Sherburne County where they're sending them to digesters. But yeah, that's a great question, and it's like also just sort of a long-stand an issue that's still going, and I think something we still need to address. Because also, I think there's also sort of a moral responsibility there too. Jill, I see you.
I'm here.
Yeah,
I'm here now.
Hey.
Yeah, what do we do with the carp? That's always the kind of a tough question. But so right now, I was just out there working with some commercial fishermen to do winter seining on a lake. And when we work with them, they do have a market that they can sell carp, which is great, because then it's actually going to be used as a food source. So that's a big part of, when there's large carp removals happening, usually it's commercial fishermen that are kind of involved. So then they get to go someplace. When we're doing smaller removals, that's always a trick, and it's trying to find places you can take them, pig farms, composting, wildlife centers, someone to the zoo this summer. So yeah, it's, always a battle trying to figure out what to do with these carp when we take them out.
Thanks so much.
Yeah.
All right, one more question. We've got Jennifer asked, did you measure nitrogen for any of this work? Curious how those trends compared to phosphorus.
That would actually, so we didn't measure anything. We entirely depended on you all for data. I imagine there's some decent nitrogen data in Equus, and we could probably try to add that in here. And I guess one thing I'm thinking we probably need to keep this work going in some way. And so I'll need to get creative about funding so that we can address some of these questions. I think the nitrogen thing is super interesting, and there's a lot we don't understand about it. And I think that's maybe where some of this, where there's overlap between managing water quality and some of the basic science questions we still have about carp management and alum treatments, for sure.
Absolutely. Okay, I am not seeing anything else in the chat. I actually did note that 9 Mile Creek Watershed District did a trial last summer with giving carp to the zoo to feed animals. So cool to know that we're working on some productive end uses for those. It is 9:59, so if there's any last minute questions, people can feel free to chime in, but otherwise I think we will wrap up our water issues presentation for this morning. Thank you so much, Jake, for spending your time with us this morning, talking about this very interesting research that you're doing, very important to the state of Minnesota. And yeah, we just appreciate spending your Thursday morning with us. And everybody have a great rest of your day.
Thanks, everybody.
Thanks, Jake. Thanks, Katie.
Really
Thanks, Jill, for popping on.
Yep, thanks.